Good morning, respected chairpersons, distinguished researchers, faculty members, and fellow participants. My name is Insha Rizwan, and I am from Delhi Technological University. And today I will be presenting our research, which is titled on Integrative in Silico Repurposing of Kinase Inhibitors for Gastric Cancer using a multi-target precision medicine strategy. This work was conducted under the guidance of my guide, Dr. Prakash Chandra, and we explored how computational drug repurposing can identify clinically relevant kinase inhibitors for gastric cancer uh, using an integrated precision oncology framework, which involved molecular docking, binding stability analysis, and admin profiling. So, despite advances in oncology, the treatment outcomes remain limited due to the tumor heterogeneity, therapeutic resistance, and activation of multiple oncogenic signaling pathways. So, several receptor tyrosine kinases, for example, particularly the EGFR, CMET, ALK, and ABL, they all play major roles in tumor proliferation, metastasis, angiogenesis, and resistance mechanisms in gastric cancer. So, traditional drug discovery is extremely expensive and time consuming, which often requires more than a decade for successful clinical translation. So, drug repurposing has emerged as an attractive alternative because the FDA approved drugs they already possess the established pharmacokinetic and safety profiles. So, an important advantage of kinase inhibitors is that they have a polypharmacological behavior, which means a single inhibitor can simultaneously affect multiple signaling pathways. This is especially important in gastric cancer because tumor evolution is highly dynamic, and even if one pathway is inhibited, compensatory signaling pathways can reactivate the tumor progression. So, coming to the aim of our study, we selected five approved inhibitors, which are crizotinib, dasatinib, erlotinib, imatinib, and venetoclax. These inhibitors were evaluated against four therapeutically relevant gastric cancer targets, which are ALK, ABL, CMET, and EGFR. These targets were specifically suggested because they are strongly associated with gastric cancer progression, metastasis, abnormal cellular signaling, and therapeutic resistance. And since gastric cancer is molecularly heterogeneous, the targeting of only one pathway may not be sufficient for effective treatment. Therefore, by selecting biologically interconnected kinase pathways, as here, we aim to simulate a more clinically realistic multi-target therapeutic strategy. Rather than just depending on docking scores, we also integrated molecular docking, cluster stability analysis, redocking validation, as well as multi-platform admit profiling, which we will be discussing further. So the methodology part, protein and ligand preparation was done basically, which included hydrogen addition, charge assignment and assessment, energy minimization using tools like Autodock and OpenBabel. The molecular docking was then performed using Autodock Vina with an exhaustiveness value of 32. Importantly, 30 independent docking runs were performed for every drug protein complex to improve the reproducibility and minimize the stochastic bias that could be seen. So, followed by docking, the RMSD based cluster analysis was carried out to evaluate the conformational stability and consistency of the binding forces. To validate the docking reliability, the native ligand redocking was performed, and the RMSD values below 2 confirmed that the docking protocol could accurately reproduce the experimental value, which means that if we did not get a value which follows this, that is a value which is not below 2, that would not confirm that the protocol could be reproduced. Finally, admit profiling was done, which we will be discussing further. So, to the docking, what were the results that we found? So the binding energies range from approximately minus 5.5 to minus 11 kilocalories per mole, which indicates that favorable target engagement was seen across several oncogenic pathways. So among these evaluated complexes, we found out that imatinib demonstrated the strongest interaction with ALK and with a binding energy of approximately minus 10.32. Whereas venetoclax exhibited the highest affinity towards ABL with a binding energy of minus 11.02 kilocalories per mole, representing the strongest interaction observed in the study. Imatinib also showed stable interaction within the catalytic pocket of c-Met, and most interestingly, crizotinib demonstrated very strong affinity towards EGFR, even outperforming erlotinib, which is clinically established EGFR inhibitor, by the way. So this was particularly significant observation because it suggests that unexpected cross-target therapeutic potential is actually there, and detailed interaction analysis shows that the presence of multiple stabilizing forces, which includes the hydrogen bonds, hydrophobic interactions, electrostatic interactions, and halogen bondings within these active sites are also present, as we have mentioned in the table. For example, we can say that venetoclax performed extensive electrostatic and hydrophobic interactions within the ABL sites. So collectively, these findings show us that substantial polypharmacological behavior among the evaluated kinase inhibitors is actually there, and this is a good evidence for that. So this multi-target interaction capability may be therapeutically beneficial in gastric cancer, where simultaneous activation of multiple signaling pathways can contribute to the disease progression and drug resistance. Now, moving on to cluster analysis and validation, several complexes form the stable conformational clusters, including the erlotinib ABL, crizotinib EGFR, and imatinib associated complexes. So, stable clustering indicates that the ligands repeatedly adopted similar binding orientation, suggesting that the reliable target engagement was there rather than random docking results that we got. And importantly, all RMSD values remain below 2 angstroms, which we said is important, with EGFR showing an exceptionally low RMSD value of 0.37 angstroms. These results strongly validate the reliability and the reproducibility of our docking protocol. Coming to the ADMET analysis, this was the last part of our evaluation, where our ADMET analysis revealed substantial differences among the evaluated inhibitors. So, for let's say example, dasatinib demonstrated the most balanced pharmacokinetic profile, including high gastrointestinal absorption, zero Lipinski violations, and absence of P-glycoprotein substrate liability. Now, this is particularly important because P-glycoprotein mediated drug efflux is a major contributor to multi-drug resistance in cancer therapy. So, erlotinib demonstrated excellent intestinal absorption and blood-brain barrier permeability on top of that, suggesting that it has potential in gastric cancer patients with CNS metastasis. Now, in contrast, venetoclax, despite having strong docking affinities, which we discussed, it showed poor bioavailability, high molecular weight, and multiple drug likeliness violations. This reduced its translational suitability as a whole. These findings clearly demonstrate us why integrating the ADMET analysis with docking studies is essential to avoid the false positive prioritization. Now, the integrated ranking. So, bisatinib emerged as the most promising candidate overall because of its balanced multi-target activity, favorable absorption profile, and acceptable metabolic characteristics. Erlotinib ranked second because of its excellent permeability and CNS penetration potential. Crizotinib also demonstrated a promising cross-target activity, particularly against EGFR, as we discussed. And meanwhile, venetoclax ranks the lowest, despite strong docking scores, because unfavorable pharmacokinetic properties reduced its overall therapeutic suitability. So, this ranking overall demonstrates that clinically meaningful repurposing decisions, they require balancing the efficacy with pharmacokinetic feasibility. So, one of the key conclusions from the study that we can draw is that strong docking affinity alone does not guarantee a therapeutic success. Clinically useful candidates, they require stable target engagement, favorable pharmacokinetics, and acceptable toxicity profiles simultaneously. Dasatinib emerged as the strongest candidate because of its balanced profile and potential ability to overcome the multi-drug resistance, which was one of the main concerns we highlighted. Allortinib demonstrated additional significance because of its blood-brain barrier permeability, which may benefit metastatic gastric cancer patients which have involvement of CNS. More broadly, if we discuss, this also supports the growing importance of a multi-target kinase inhibition strategy, which we need to look into. And we can't, of course, forget venetoclax, which showed us that despite having the strong docking affinities, the poor bioavailability and unfavorable drug likeness reduced its overall clinical suitability. So the conclusion, among the evaluated candidates, like we said, dasatinib emerged as the most promising candidate for an inhibitor due to its stable multi-target interactions, favorable ADMET profile, and reproducible binding behavior. Allortinib demonstrated the additional therapeutic potentials for CNS-associated gastric cancer patients, and this also highlighted that docking affinity is insufficient for candidate prioritization without pharmacokinetic integration. So overall, our work highlights the growing importance of integrated in silico precision medicine approaches in accelerating oncology drug discovery and drug repurposing. By combining the computational efficiency with translational relevance, these strategies may help us to bridge the gap between molecular discovery and personalized cancer therapies. Thank you so much for your attention. I would like to acknowledge my guide, collaborators, and the conference organizers for providing me with this opportunity to speak on an international platform. Thank you so much.